easy to understand the things. So that's why the most of the research were carried out on the prokaryotes. Find or the ultra-structure of the ribosome is a very complex structure. In it, the RNA and proteins are intervened and arranged in a complex manner in two subunits. Two subunits we have seen. One large unit it is there, and another small unit it is there. Since the positive protein charges are not sufficient to balance the many negative charges in phosphates of the RNA, so ribosomes are strongly negative and bind cations and basic strains. Consequently, negative staining of the ribosomes has led to better understanding of the fine structure of these granules in these organelles. Actually, this is the experimental part that how these ribosomes they were stained with the different stains, and then they were studied under the different microscopic conditions. Recently, it is found that the two models have been suggested to explain the three-dimensional structure of the prokaryotes, and that is the 70S ribosomes. Stoffler and Whitman model. This is also called as a quasi-symmetrical model, which is proposed in 1977. According to this model, the 30S ribosome subunit has an elongated, slightly bent prolate shape, which is bipartite structure. This is the structure. This is two lobes were present. This is the 50 subunit, this is the 30 subunit, and they have certain kind of projections. And these projections they were called as the cleft and the dent. The head it is there on the upper side. This is the main body. Then the central protuberance, means the central extension it is there, and this is the evaluated seat. Actually, this part is mainly responsible for the production of the proteins, and this is the base, and this one is the tunnel. That we will also discuss when we will discuss different kind of protein synthesis. So this is the Stoffler and Whitman's model of the 70S ribosome. The transverse hollow or a cleft divided the 30 subunits into two parts: smaller head, larger body. Head it is the smaller part. The larger is this one is the body. This unit, this 70S, and the base unit is the 50S. So it is further divided into the two parts. The head it is there, and then the central body it is there. Then 30S and 50S subunits become associated, and they form the 70S ribosome. Lake's model, this is the asymmetrical model which was proposed in 1981. This is a completely asymmetrical model of ribosomes, and it has been suggested by the James A. Lake in 1981. So the Lake's model of 70 ribosome note that the three-dimensional structure of the ribosome in two different organizations. This is only in the two forms. First part it is there, and this second part it is there. And the base it is there, and two models are there: A model and the B model. So again, these two units, the smaller subunit and the larger subunit, they were joined together and they formed the structure like that. Similarly, in this case, in B form, the head it is there, the valley it is there, the both sides, the ridge is there, the central protuberance is there. When they join together, and they form this kind of structure. Smaller subunit and the larger subunit, they were joined and they formed the ribosome. The smaller subunit has a head, base, and platform. The platform separates the head from the base by the help of the cleft. This cleft is an important functional region, suggests to be the site of the codon or anticodon interaction. As we have seen, we have studied regarding the protein triplet of the codon were formed, and which is responsible for the production of particular proteins or the required proteins. So this cleft region is this actually this is the cleft region. This region is responsible for the recognition of the different codons, and these codons they were responsible for the production of the different proteins. So these codons, anticodon interaction, and has a part of the binding site for the initiation factor of the protein synthesis. The larger subunit consists of the ridge, central protuberance, and the stalk. The functions of the ribosome, the main function of the ribosome in the cell is the protein synthesis, as the ribosome has two subunits. So the smaller 30 subunits bind to the mRNA and reach the code present in the mRNA. Yeah, this mRNA we have seen that this mRNA or the messenger RNA, which were formed in the nucleus, and it moves from nucleus, from inside the nucleus to outside or to the cytoplasm, and they found these ribosomes, and from these ribosomes the codes and anticodons were joined together, and they formed the long polypeptide chain, and these polypeptide chains were converted into the particular proteins. So when the messenger RNA reaches, what happens? The 30 subunits they bind to the mRNA. Firstly, both the units they were not joined together. When the 30 subunits join together to the mRNA, and then it reads the code present in the mRNA, the larger 50 subunits receive the amino acid bound by the tRNA and link them together, thus forming the polypeptide amino acid chains, that is the proteins. Ribosomes also have a protective function. mRNA stand between the two subunits of the ribosomes, is protected by the action of the nuclease enzyme. Similarly, the nascent polypeptide chain passing through the tunnel or the channel between the subunits are protected against the action of the different digestive enzymes. Another cell organelle which is included in your course, that is the endoplasmic reticulum. I think before endoplasmic reticulum, another part it is also there, I think, the Golgi complex. That we will discuss on the next turn. So right now we can start the endoplasmic reticulum. Endoplasmic reticulum, which is denoted by the ER, is a eukaryotic organelle that forms the interconnected networks of the tubules, vesicle, and cisternae within the cell. This endoplasmic reticulum was discovered by the K.R. Porter, Claude, and the Thomson in 1945, but the name has been given by the Porter in 1953. So, as we have seen in case of the cell theory, the number of workers have carried out the different experiments, and according to their thought, according to their imagination, and they have given the name in different ways. But finally, the cell name was given by the different workers. So similarly, during the study carried out by the different workers, like in case of the endoplasmic reticulum, K.R. Porter, Claude, and Thomson, they discovered basically in 1945 that is the endoplasmic reticulum, but the name has not been given at that time. The proper name, like endoplasmic reticulum, was given by the Porter in 1953, means about 8 years after its discovery. According to the Dalmer, 1966, endoplasmic reticulum is originated from the plasma membrane by the investigation. According to the D. Ropartis, 1970, the endoplasmic reticulum originates from the nuclear envelope. That we have also seen in certain figures, that from the modification of the nuclear envelope, some projections were there, or we can say the layer of projections were there, then sorry, that projection is called as uh, called as the endoplasmic reticulum, and also the presence of the ribosome denotes the characteristic feature of the rough endoplasmic reticulum. This is the diagrammatic representation. If you recall the nucleus, then you can say that this is the part of the nucleus, a very small part of the nucleus, which contains the nuclear core complex and the dot-like structure. These are the ribosomes. So from this nuclear envelope, certain kind of the layer of the projections were there, and these projections they were called as the endoplasmic reticulum. Even in this endoplasmic reticulum, you can see there's certain areas they composed of the dot-like structures. These are the ribosomes. So this is converted. This is called as the rough endoplasmic reticulum. But at the same time, if you can see, certain projections were also there, and these were without the presence of the ribosomes. Then they were considered as the smooth endoplasmic reticulum. And now, what are the cisternes? What are the vesicles that we will discuss in the next slide? So, the endoplasmic reticulum occurs in all eukaryotic cells except ova, embryonic cells, and mature RBCs. It is also absent in prokaryotes. Endoplasmic reticulum is quite extensive in metabolically active cells, simple in storage cells, and reduced in spermatocytes. By this particular point, you can say that according to the function of the cell, is the number of its activity depends. If the cell is very active, it is participating in certain kind of activity, then uh, the cell is active, and it depends on it depends the number of the ribosomes or the type of the endoplasmic reticulum is present. So in the active cells, it is quite extensive, but if the cell is inactive, then it is in the form of the storage, and it is reduced in the spermatocyte, where these are almost not required. Endoplasmic reticulum is a strikingly complex structure within the cell and has three morphological distinct regions. So, as we have seen, that the endoplasmic reticulum is also a morphologically contains the morphologically different regions. As we have seen in case of the ribosome, that two subunits were present. Even one subunit is also composed of the different regions, like head it is there, cleft it is there, the body it is there, and the central area it is there, which was responsible for the reading of the codons and anticodons. So similarly, in case of the endoplasmic reticulum, the morphologically different regions were present in case of the endoplasmic reticulum. The general structure of the endoplasmic reticulum is an extensive membrane network of the cisternae. This is called as the sac-like structures. They were held together by the cytoskeleton. The phospholipid membrane of the endoplasmic reticulum encloses a space, the cisternae space or the lumen from the cytosol. The endoplasmic reticulum is an extensive three-dimensional structure of the vacuolar system that extends from the nuclear membrane up to the plasma membrane. Endoplasmic reticulum occupies about 10% of the total cell volume, that we have also discussed in the previous classes. That the total of the volume of the cell, 10% is composed by the endoplasmic reticulum. And why it is endoplasmic reticulum, you can see, because most of the enzyme or most of the proteins which are going to be performed, they are going to be performed with the help of the ribosomes, and the ribosomes were present on the endoplasmic reticulum. As we know that proteins are the main part, we can say the chemical part of the cell, because whenever the enzyme is going to be formed, then it will be modification of the different proteins. So its membrane accounts about 30 to 60% of the cellular membranes, and increase the internal surface 30 to 40 times as compared to the external surface. Endoplasmic reticulum divides the intercellular space into two distinct compartments, like luminal inside the endoplasmic reticulum and extraluminal cytoplasmic compartments were there. Then endoplasmic reticulum exists in three morphological forms: cisternal, tubules, and vesicle. All of them are filled with the fluid, and they are called as the endoplasmic matrix. So, if we recall the membrane of the nucleus, then we can say that it was the bilipid layer, biphospholipids. Two layers were present, and in between the two layers, the perinuclear space were present. But here there is no any space were present. Whatsoever the space is present, that is filled with the certain kind of fluid that is called as the endoplasmic matrix. So, if we consider the cisternae, then we can say that these are the flattened, unbranched sac-like structures. Uh, Flattened structures were there like that, and on that, the different dot-like structures were present. They were called as the ribosomes. They lie parallel to each other. The sac-like structures, they were present in the parallel fashion, and they are interconnected and may be converted with the may be covered with the ribosomes. ये जो सिंपल सा आपको बहुत सारी बुक में दिखेगा, ये इस तरीके का इतना सिंपल नहीं होता है. They are interconnected. ये जुड़े हुए होते हैं कहीं ना कहीं से, and they occur in the cell having the active synthetic role. Then the tubules, these are irregular branches, tube-like structures which form the network along with the other elements. एक वो simple plate-like structures थी जो parallelly attached हैं with certain kind of structures. But here कोई एक particular ऐसा function नहीं है, but irregular branching system है tubes का, and even on that the ribosomes, they were present. They do not possess ribosomes on their surface. In certain cases, it was found that the ribosomes were present, but mostly they were, as we have seen in the previous slides, the ribosomes were absent, or they were called as the smooth endoplasmic reticulum. They are more common in the cell, engage in lipid and the steroid synthesis. So just you can see the difference. जहाँ पर प्रोटीन की या सेल को बहुत ज्यादा काम करना पड़ रहा है, वहाँ पर या प्रोटीन और एंजाइम की जरूरत पड़ रही है, तो वहाँ पर राइबोसोम्स ज्यादा प्रेजेंट हैं, और वो किस तरीके से प्रेजेंट हैं, वो वहाँ काम कर रहे हैं. जहाँ पर लिपिड की सिंथेसिस हो रही है या स्टेरॉइड की सिंथेसिस हो रही है, तो वहाँ पर राइबोसोम्स की जरूरत नहीं है. तो these functions were carried out by the different type of the tubules, different structure of the endoplasmic reticulum. Then the vesicles, these are the round, spherical, or ovoid structures. The diameter of the vesicles, which is ranges from 25 to 500 micron, and they are found dispersed in the cytoplasm. तो एक आपका अटैच है, दूसरा जो है वो ब्रांच फॉर्म में है, और तीसरा जो है वो अवॉइड स्ट्रक्चर्स हैं, ओवॉइड और स्फेरिकल स्ट्रक्चर्स हैं जो फ्री फ्लोटिंग है आपके साइटोप्लाज्म में. They are rich in pancreatic cells, in spermatocytes. Vesicles are the only structure found in spermatocytes. When the cells are disrupted during the cell fractionation, the endoplasmic reticulum breaks up into the small vesicles, and these are called as the microsomes. Then types of the endoplasmic reticulum. There are two types of endoplasmic reticulum: SER, smooth, or the agranular reticulum, and the RER, this is the rough or the granular endoplasmic reticulum. Granular and agranular depends on the presence and absence of the ribosomes. If they were present, then they were called as the granular. If they were absent, then they were called as the agranular. Smooth or agranular endoplasmic reticulum. The endoplasmic reticulum, in absence of the ribosomes, appears smooth and called as SER, composed of the many tubules and vesicles. This agranular reticulum is most common in those cells which are concerned with steroid or lipid synthesis, such as the adrenal and sebaceous glands, gonadal interstitial cells, then the carbohydrate metabolism, liver cells, electrolyte excretion, as in case of as in the chloride cells of the fish gills, impulse conduction in muscle cells, and with the pigment production, as in retinal pigment cells, or that is the non-protein type of the synthesis. Then rough or the granular endoplasmic reticulum, they were characterized by the presence of the ribosomes, and they contains two types of the proteins, riboforin 1 and second, attached to the ribosomes. Rough endoplasmic reticulum composed of the many cisternae tubules are very few. So depend on the rough and the smooth, their structure different. If it is if it is rough, then the number of cisternae or number of sac-like structures were arranged parallel. That was found in the case of the rough endoplasmic reticulum. Tubules are in a very few number. Rough endoplasmic reticulum predominates in the cells which are actively synthesizing proteins such as enzymes and in glands. That I said that depending on the type of the cell or which type of the function is carried out by the cells. If it is very active, the number of enzymes and proteins were continuously formed, then the endoplasmic reticulum presents shows that they were present in the cisternal form. Out of the total endoplasmic reticulum content, about two-thirds is rough endoplasmic reticulum and one-third is smooth endoplasmic reticulum. Smooth and rough endoplasmic are interconvertible. Interconvertible, but whenever the cell is required or there is a signal that certain kind of protein and enzyme have to be formed, then the cell become active and a smooth endoplasmic reticulum can be converted into the rough endoplasmic reticulum as the appearance of the ribosomes occurs, or when the cell is going from active form to the inactive form, then this endoplasmic reticulum converted into the smooth endoplasmic reticulum. So it depends on the type and the situation of the cells. Rough endoplasmic is more stable than smooth endoplasmic reticulum. Rough is basophilic due to the presence of the ribosomes. The function of the endoplasmic reticulum provides the mechanical support to the cytoplasmic matrix. It also provides the large surface area for the synthesis of the various materials, as I said, that the proteins, enzymes, and certain other material, lipids, and steroids, etc. Then endoplasmic reticulum keeps the various organelles in their position due to their structure. They can be found in a particular fashion, and they also help in the maintenance of the position of the organelles. Endoplasmic facilitates the quick intracellular transport, thus forming the circulatory system of the cell. Then it extends through the plasmodesmata and as desmotubules and controls the movement of the material between the adjacent cells. So not only it is performing in within a cell, but sometimes it can extend through the plasmodesmata, or these are the cytoplasmic connections between the two cells, and can help in the movement of different materials from one cell to the another cells. And these plasmodesmata movement, extension of this endoplasmic reticulum through the plasmodesmata, it's called as the desmotubules. Then the function of the endoplasmic reticulum, again, the rough contains the ribophorines for holding the ribosomes. Then rough endoplasmic reticulum synthesizes proteins destined for the secretions, lysosomes, or the plasma membrane. Then and the rough endoplasmic reticulum forms the nuclear envelope, plasma membrane, and the smooth endoplasmic reticulum. Then the function of the smooth endoplasmic reticulum, it takes part in the synthesis of the lipid, steroids, hormones, ascorbic acid. Then it also helps in the detoxification of the toxins using cytochrome P450. In liver cells, rough possesses enzyme bodies called as the glycosomes for glycogen metabolism, like glycogenesis and glycogenolysis. In retinal cells, rough smooth produces the vesicle pigment from the vitamin A. In muscles, smooth endoplasmic reticulum modified into the sarcoplasmic reticulum (SR), which stores and releases the calcium ions, forms the muscle contraction. They were formed during the muscle contraction. Then this smooth endoplasmic reticulum also produces organelles like Golgi complex, Golgi apparatus, lysosomes, spherosomes, and the vacuoles. Modification of the endoplasmic reticulum, annulate or annulate lamellae. This was described by the McCouch in 1952. These are the endoplasmic reticulum having pore-like nuclear pores. They form from blabbing nuclear envelope. They form to the nuclear envelope, and they can be smooth or rough. Then sarcoplasmic reticulum, modified form of the smooth endoplasmic reticulum is striated muscle cells, both skeletal and cardiac. They supply the calcium ion to the muscle cytoplasm, help in intracellular impulse transmission and contraction. Then nasal granules, these are called the tergioid bodies. They are the masses of the RER, rough endoplasmic reticulum, in cytone of a neuron. Then ergastoplasm. This term was introduced by the C. Gamier in 1900 for highly developed endoplasmic reticulum with ribosomes in synthetically active cells. Thus, ergastoplasm refers to the well-developed endoplasmic reticulum along with the ribosome in some part of the metabolically active cells. So, as I said, that whatsoever the activity which is going on the cell, or whatsoever the activeness of the particular organelle depends on the type of the cell on which, or we can say that the which different function is going to be happen, or which different function that particular cell is going to be performed. If it is in active form, then all the other organelles are also in the active form, and they are performing the different functions. Then another form is the myeloid bodies. This is also the specialization of the granular endoplasmic reticulum. These bodies occur in pigmented epithelial cells of the retina. Each body consists of the vesicles and tubules packed together near the basement membrane of the cells. Ribosomes are not associated with these bodies. Each myeloid body is biconvex, 4 to 5 micron long, along with the axis, and they were composed of the stacks of the packed lamellae. Packed lamellae means whatsoever we have seen in case of the cisternae. Then the lamellae were present, and they were joined together with the particular structure. Then the three varieties are the rough endoplasmic reticulum, smooth endoplasmic reticulum, and sarcoplasmic reticulum. In case of the rough, due to the presence of the ribosomes or due to the work carried out like the protein manufacturing, protein functioning, then smooth, this will help in the production of the different steroids and lipids. Then the sarcoplasmic reticulum from the this word sarco, sarx. This work have been derived from the Greek word sarx, that's the flesh, and that is the special type of the smooth endoplasmic reticulum found in the smooth and striated muscles. The function of these particular structures is it plays an important role in the production, processing, and transportation of the protein and lipid molecules. Endoplasmic reticulum produces the transmembrane proteins and lipids for its membrane, as well as the membrane of the lysosome, secretory vesicles, Golgi apparatus, cell membrane, and the plant vacuole. So the endoplasmic reticulum is uh, playing a very important role, not only in the production of the different kind of proteins and lipid molecules, but they are also helps in the maintaining the different membranes of the lysosome, secretory vesicles, Golgi apparatus, cell membrane, and even the plant vacuole. So this is all about our endoplasmic reticulum, and now I can stop here because if we start our new topic, then it will take slightly more time, and you may have some different classes. So I request all of you that I have posted the notice regarding the um, submission of your assignment, and do remember that it is composed of the 70 marks. It carries the 70 marks. 70 marks will be the total marks which you have to submit the assignment for two different MEMS also, Dr. Wasia Basri and Dr. Meher Fatima. They were also going to conduct a certain kind of assignment or some sessional tests. Whatsoever mode they apply, I'm not going into the details, but they were also going to take the sessional, and we will compile the whole marks. My assignment, Dr. Wasia assignment, or the